Hello guys, welcome back to the channel. Today we are going to discuss meiosis, but now it's going to be in two parts because I'm trying to avoid making it giving you a very long video, one video, but it's better if they become two videos so that it's going to be easier. Now, during part one, you will see what we're going to talk about, and then there's going to be continuation during part two. Now, before we start, don't forget to like, don't forget to subscribe to the channel, don't forget to share, and last but not least, don't forget to comment down below. Suggestions are also allowed. Now, let's start by looking at what we are going to discuss. Okay, now I'm going to give you the description of the entire lesson, even though it's going to be in two parts. Okay, I'm going to post these two parts at the same day, so don't stress. Now, when looking at the lesson plan, we're going to define meiosis, which is what we always do. We have to always define the topic that you're going to discuss, because you need to know what the topic is about. And then we're going to look at the site of meiosis in animals, or the place where meiosis occurs in animals. We're going to look at the events of interphase, or we're going to just look at interphase, which is a phase that comes before meiosis. Okay, it's interphase because it's between the two phases, mitosis and meiosis. And then we're going to look at the events of meiosis 1 and 2. We're going to look at the importance of meiosis. We're going to look at the abnormal meiosis. We're going to look at we're going to compare the two, mitosis and meiosis. And last but not least, we're going to look at the karyotype. Now, what is meiosis? In simple terms, meiosis is a type of cell division whereby a diploid cell undergoes two divisions, which are 1 and 2, division 1 and 2, and then it divides to form 4 unidentical haploid cells or sex cells. Now, what are diploid cells? What are haploid cells? Diploid cells have two sets of chromosomes where each chromosome has a homologous partner. They have partners. Homologous, I'm going to explain what that means. And then haploid cells have only one set of chromosomes and they do not have homologous partners. So meiosis is divided into two phases, divisions 1 and 2, meiosis 1 and meiosis 2. Where does it occur? Where does meiosis occur? It occurs in what we call gonads or sex organs. It occurs not sex organs, It occurs in gonads whereby gametogenesis occurs. So in females, it occurs in ovaries, and then in males, it occurs in testes. And as I've stated during the description of the lesson, we are going to look at a phase that comes before meiosis, which is referred to as interphase. So before meiosis occurs, or before it starts, or during interphase, DNA replication has to take place. I discussed DNA replication in my previous video. I'm not going to repeat it. If you want more information on it, it's better you go watch that video. And then this DNA replication, it leads to, or it results in two sets of chromosomes, which consist of two identical chromatids joined together with a centromere. What is a centromere? What is a chromatid? I'm going to explain. Now, first, I need to explain to you what is homologous, because as you can see on our diagram there, there's what we call homologous pair of chromosomes. This is from the father, this is from the mother. Okay? Now, what does homologous mean? Homologous simply means that now these chromosomes, they're similar in size. They have the same size. Okay? They have the same gene, same type of gene, okay, which is located at the same place. What I mean by the same type of gene, it might be gene for eye color, gene that is coding for eye color, or gene that is coding for hair color or hair texture, and so forth. And as I've stated, that during interphase replication has to occur. So this chromosome has to make a copy of itself. Okay, now once it makes a copy of itself, that copy of itself is joined to the original one, by what we call a centromere, which is at the center here. This structure at the center, unfortunately, is similar in color with the chromosome, the sister chromatid, it's fine. But now this structure at the center here, it joins these two sister chromatids. What is sister chromatid? Sister chromatid, it's what is formed. It's a copy of the original chromosome. You know, it's a copy of the original chromosome. So individually, they are called, once now they are together, joined by a centromere, they are called sister chromatids. But this is still a chromosome from the father. This one is from the mother. So both chromosomes from each parent, they will undergo replication to form sister chromatids. But still, this is also referred to as a chromosome because it's formed from the original, this one. So it's the same thing, but now in different, in pieces. It's a copy of this one, and it's joined together by a centromere. It's really a bit complicated, but not, yeah, it's really a bit complicated, but just know that the chromosome from the mother has to undergo replication to form what we call sister chromatids, which are going to be joined together by a centromere. Now, as I've stated, meiosis consists of two cell divisions, meiosis I and meiosis II. So what happens during the first meiotic division or meiosis I is that the number of cells is doubled, meaning one cell will form two cells. But the number of chromosomes in the cell, they're not doubled. Instead, they result in half as many chromosomes per cell, which is something you're gonna see in diagrams as we continue with this lesson. And then in meiosis number 2, or the second meiotic division, the number of chromosomes does not get reduced. Okay, but now what happens is that now 4 cells are going to be formed, and these cells at the end, they're going to be unidentical. They're going to be different from each other genetically. Okay, now the first phase of meiosis I and meiosis II, they are called the same names, but what makes them different is the numericals, the numbers. You need to be specific with which phase of which meiosis we're talking about. Okay, so now looking at meiosis I, and the first phase of meiosis I is what we call prophase I. So what happens during prophase I is that the chromosomes, they start to become shorter, or they shorten, and then as they become shorter, they also become visible. Okay, it means they become condensed. That's what it means by shorter. They become condensed, and then they start to be visible. As you can see there, then they appear as two chromatids joined by a centromere. And then homologous pairs of chromosomes are now visible. I explained what homologous means, but let me repeat. Now, looking at your diagram there, you see these two chromosomes, 1 and 2, they form a homologous pair. Why? This chromosome and this chromosome, they're of the same size. You can see they're long. Both of them, they are long. They're of the same size, and they carry same type of gene. It might be maybe gene for eye color, and those genes are located at the same place. As you see, even the other ones, they are short, now, and they also have similar genes, type of genes, and those genes are located at the same place. Now, what also happens during prophase I is that the nuclear membrane and the nucleolus, they will start to disappear. A nucleolus is like a nucleus within a nucleus. They will start to disappear, And then now the spindle fibers also, they start to form. Okay, as the other things disappear, spindle fibers form. Okay, and then there's a very important process that's going to occur, which we refer to as crossing over here. Crossing over is when now the chromatids of each homologous pair, note that a chromatid of each homologous pair, they touch, and they touch at a particular place or at a particular site. That site, we call it a chiasma or a chiasmata. Some call it a chiasmata. Okay? Now, as they touch, something very special occurs there. They touch because of the fact that they're trying to exchange information. But now, which information? Genetic information or genetic material to bring about a variation. Okay? And then as that occurs, as the crossing over still occurs, what happens? More of the spindle fibers are continued to form. That's the end of Prophase I. So for Prophase I, what is important is that now the chromosomes, they become visible, especially the homologous pairs of chromosomes, they start to be visible. The spindle fiber starts to form. The nuclear membrane and the nucleolus, they start to disappear, and crossing over has to take place. And the spindle fibers also continue to form even further during Prophase I. Now, moving to the next phase of meiosis I. Now, this is what we call metaphase. During metaphase I of meiosis I, what will happen now? The spindle fibers, they will start to extend across the whole cell, as you can see on the diagram here. The spindle fibers, they extended across the whole cell. Now, the homologous chromosome pairs, they line up along the equator, you know, they line up in pairs, in homologous pairs. You see, the long chromosomes, they're still together at the same position, connected to what? Connected to these spindle fibers. The short ones, the same thing occurs to them. They are in pairs as well, homologous pairs. And then what you need to notice is that now, the centromere of each chromosome, they're still attached to their they're attached to the spindle fibers. That's it. What is important for metaphase is that now these chromosome pairs, metaphase I specifically, these homologous pairs, they line along the equator in pairs, in homologous pairs. That's what is important. And also what is important is that now these homologous pairs, as they line up along the equator, there's what we call random segregation or independent assortment that occurs. They don't have to follow a specific procedure to be lined along the equator, but they randomly line up so that during the next phase, when they separate, they randomly separate. No specific ones are supposed to go to the other cell, or other specific ones are supposed to go to the other cell. No, they randomly go to each part of the cell. And then that part of the cell is divided into two cells. It forms two cells. And that stage is what we call Anaphase I. So during Anaphase I, what happens now? These chromosomes that are in pairs now, in homologous pairs, during Anaphase I, they are getting separated from each other. They are pulled towards the poles. They are no longer a pair now. The pair is separated. The partners now, they are separated. They are getting divorced. They are separated from each other. One moves to the right, one moves to the left. So the homologous pairs are now broken up. Okay? And I want you to notice here that As much as you remember, during anaphase in mitosis, what happens is different from anaphase I of meiosis. Here, what happens is that in anaphase I, the homologous pairs are separated, meaning now a chromosome, an entire chromosome will move to opposite side of the poles, and other one move to the opposite side of the pole, an entire chromosome. But now during mitosis in anaphase, what happens is that now in metaphase, during mitosis, individual chromosomes, they line up along the equator, and chromatids during anaphase during mitosis, they are becoming separated. So a chromosome becomes separated into chromatids during mitosis. That's what I'm trying to explain. Now, looking at the last phase of meiosis I, which is called the telophase. During the telophase, what happens is that the chromosomes reach the poles of the cell. Once they reach the poles, what will happen is that now each pole will have half the number of chromosomes present in the original cell. And then cytokinesis will occur, meaning now the cytoplasm will divide as the cell membrane constricts. And then two cells with half the number of chromosomes are formed. And then that's the end of meiosis I. And then now there's going to be the beginning of meiosis II. Now in meiosis II, we are taking over from telophase I, the very same cells that resulted at the end of telophase I, they are going to be taken to meiosis II. Now, because now we have got two cells, what happens in the cell on your right happens on the cell on your left. Similar processes occur in each cell. Okay? Now, the spindle fibers in each of the new cells are still there. They're still formed. They're still okay. Okay? Now, we move to the second phase of meiosis II, which is Metaphase II. Remember, during metaphase, something has to line up along the equator. But now during metaphase II, what lines up is the individual chromosomes. Metaphase II is different from one because during metaphase I, it's homologous pairs that lie along the equator. But during metaphase II, it's only the individual chromosomes that align are lining up along the equator. Okay, and they're still the spindle fibers, they're still attached to the centromeres. Now moving to the next phase of meiosis II, which is Anaphase II. During Anaphase II now, something similar to metaphase, to anaphase of the mitotic or mitosis division, it occurs. What happens now is that now the chromatids are now pulled to opposite directions towards opposite poles. So Anaphase II of meiosis is similar to anaphase of mitosis. Okay, now what pulls these chromatids towards opposite poles is the spindle fibers, because now they contract. And then as they contract, they pull these chromatids to opposite poles. Now, once that occurs, the next phase results, which is telophase II. Then during telophase II, now these daughter chromosomes or chromatids, they reach the opposite poles. And then what happens? The nucleus starts to form. The nucleus will start to form. The membrane will start to form. You know, the nuclear membrane, so not the cell membrane though, but the nuclear membrane. Most of the things that disappeared in the beginning of the division start to form again. And then what happens is there's going to be cytokinesis. meaning the cytoplasm will divide as the cell membrane constricts, which is referred to as cytokinesis. So after cytokinesis has occurred, we have got 4 haploid daughter cells that are formed. And then these cells that are formed are not the same. They're genetically different, very different, because of genetic variation that was introduced because of crossing over and random segregation. Okay? That's it. 4 haploid daughter cells are formed. What are haploid daughter cells? Are cells that contain only one set of chromosomes. You can see one set of chromosomes, and they are different from each other. And then that's it. And then now the easiest way to remember meiosis is by using IPMET or IPMET. It's up to you how you say it, but there's a point that I want to emphasize is that interphase is not part of meiosis, and it's either not part of mitosis, but it's just a phase that is between these two. It's just a phase that is between these two cell divisions, mitosis and meiosis. I just want you to know it because sometimes in the exam, you will be asked what comes before meiosis, and then you have to say it's interphase. Okay? Then they will ask you what's the first stage or the first phase of meiosis. Then that's when you say prophase. The first stage or the first phase of meiosis is always prophase, just like mitosis. Interphase comes before meiosis. It's just a stage or a phase between these two cell divisions, mitosis and meiosis. Okay? And then the easiest way to know what happens during prophase, P stands for preparation. You are preparing. Okay? It's preparation. The chromosomes now, they start to prepare for meiosis. Okay, they become more shorter, they become visible, and then crossing over also takes place, meaning they exchange genetic material. And then that's it for prophase. And then for metaphase, what happens now? But remember, prophase is only prophase I, whereby now there's crossing over taking place. In prophase II, we don't have crossing over taking place. Prophase II, we're preparing for the second meiosis, the mitotic division, which is meiosis II. So prophase I is the first stage. So the prophase II is the first stage of meiosis II. Okay, now metaphase. How to remember metaphase? It's like the word metaphase starts with the letter M. So M stands for middle. So the chromosomes, they are going to be lined in the middle of the cell along the equator. That's it. And then anaphase stands for apart or away, meaning now either chromosomes are going to be pulled away from each other or apart from each other, or either chromatids are going to be pushed away from each pulled against away from each other, just like that. And then telophase stands for terminal. Terminal means final, the final phase of meiosis I and meiosis II. Thank you guys for watching. I'll be bringing you the next part or the next lesson on meiosis, where we're going to look at the importance of meiosis. We're going to look at the abnormal meiosis. We're going to look at the karyotype. So please watch this video after this one so that you understand the entire topic properly, so that you are ready for the exam. Thank you for watching. Please don't forget to like, share, and subscribe. Thank you so much for lending me your ears.